Hello, everyone, and welcome. We'll wait maybe just one more minute as people are continuing to log in, and then we'll get started. All right, welcome everyone today to our OTS webinar series. Today we are featuring Professor Willeke van Roenmaam, who is a professor in the Department of Human Genetics at the Leiden University Medical Center in the Netherlands. The topic of her research is autosomal dominant neurodegenerative diseases that have aberrant protein aggregation as a pathological hallmark. Her work is highly translational in nature, working in close collaboration with clinical departments as well as biotechnology and pharmaceutical companies. This research has resulted in various published patents, some together with companies and some licensed to companies, as well as industry-sponsored research projects. Unique patient-driven fundraising initiatives contribute not only financial input, but also patient perspective to research programs in her group. Originally, her main expertise was in Huntington's disease, a brain disorder caused by a polyglutamine expansion in the Huntington protein. However, her group is now also focusing on other polyglutamine disorders, as well as a very rare disease caused by a mutation in the APP gene called HCHWA/D, hereditary cerebral hemorrhage with amyloidosis Dutch type. For all these disorders, she studies the molecular disease mechanism, identifies biomarkers, and then uses this knowledge to develop novel therapies. Using cell models, animal models, and patient materials, she applies and develops a wide range of molecular, genomic, and biosemantic assays. Most recently, she started using human-induced pluripotent stem cells as a model system. This is such an important model system for brain research at the LUMC that they've set up an iPSC neurodifferentiation lab in the department that facilitates the use of these cells for other researchers, providing technical support and standardized protocols. She's the co-founder and co-lead of the Dutch Center for RNA Therapeutics that aims to develop RNA targeting therapies for patients with ultra-rare mutations. So we are incredibly pleased to welcome you, and we just want to begin the webinar by thanking you very much for your contributions, and we're looking forward to your talk. So take it away.
Thank you very much. And I think you get extra points of saying out loud the HCHWAD abbreviation. So well done on you. So thank you very much for inviting me to give this webinar on the development of bespoke RNA therapies for patients with unique mutations. And I think it's important to highlight the global differences that we've encountered while we started up to bring these bespoke therapies also to patients in the Netherlands and in Europe. So with that, I think we all know about this landmark paper about the development of Milasen, where within 1 year after genetic diagnosis, an antisense oligonucleotide therapy was designed. And the first treatment started within a year. The reason why this was so successful and was successful in improving the quality of life in Mila was that she had really severe epileptic seizures. And if you look here, the size, the higher the dot, the more seizures she got per day, and the darker the dot, the longer the seizures were. And you can see that over the treatment period, the number of seizures decreased, and also the length of the seizures. So for her, this was a really improvement in the quality of life. And I think this really brought home that for those of us who have been developing these antisense oligonucleotide therapies for years, together with companies, for larger groups of patients, that we could use this knowledge also for single patients. And so actually in the United States, quite a few patients have already been treated, single patients with unique mutations, but so far none in Europe. So what if a patient that has, for instance, ataxia, because that is one of the diseases that I'm interested in, is caused by a mutation that is found in less than 5 patients worldwide, and if it's such a rare disease that there is no company that would be interested in developing a therapy, because it would not be possible to get a return of your investment. And it's also not possible to do a clinical trial. And in Europe, it's not even possible to get these drugs registered in the European Medicine Agency. And so that is really why we decided in the Netherlands, when we saw the paper by the group from TMU, Annemieke Aartsma and me said, we have all the technical know-how and the expertise to also do that in the Netherlands. So we first wanted to start the LIDA Center of RNA Therapeutics. But at the same time, we were also working together closely with Rob Collin from Radboud UMC, who is working on eye disorders, and Ipe Elgersma, who is working on neurodevelopmental disorders. And then we decided to make it a Dutch Center of RNA Therapeutics, because these patients are so rare, and we really wanted to group and be more efficient in implementing this in the Netherlands. So it's made up of 3 university medical centers at the moment, It's a non-for-profit consortium. And we have decided to use our expertise in splice modulation. So we are only at the moment focusing on splice modulating antisense oligonucleotides, focusing on the eye and the central nervous system, because that's where you can get a local delivery. So currently 16 approved oligonucleotide drugs with different modalities and different target tissues. And as I said, the target tissues that we are focusing on are eye and brain, and we are using splice switching antisense oligonucleotides. So I'm quite sure the audience is quite knowledgeable on splice modulating antisense oligonucleotides, but I'm just going to give you a short recap. So in the DNA, you have the coding and the non-coding paths. The pre-mRNA also includes these introns and these exons, and then through a process of splicing, the introns are removed, and then this mRNA is then translated into a protein. And why we have chosen for these splice modulating AONs, because they make use of stable FDA and EMA approved chemistries, and their toxin safety profile is quite stable. So splice modulation comes in different flavors, and what does that mean? It means that different types of DNA variants, they require different types of splice modulation, or you can apply different types of splice modulation. So there can be variants that disrupt the reading frame, and if you then design, and the disruption of this reading frame means that there is no protein expressed. If you can remove this exon by designing an AON that targets that exon, you can restore the reading frame of the mRNA, and then you can get a truncated protein expression. Another application of splice modulation, if there is a toxic gain of function in a DNA, where if it is expressed in the protein, it makes this protein toxic, so a toxic gain of function in the protein. If you can remove this exon from the mRNA and you can keep the mRNA in frame, then also you can get sort of an internally truncated protein where the part of the protein that induces this toxic gain of function is removed, and thereby you will produce a non-toxic protein. And so I'm going to show you an example of this approach of research that has been done in my group on spinocerebellar ataxia type 3. And the type of mutations that we are focusing on first in the Dutch Center of RNA Therapeutics are the deep intronic variants. So if it could be that this deep intronic variants means that there is part of the intron that is aberrantly included in the mRNA as a cryptic exon. By this inclusion of this cryptic exon, the mRNA is out of frame, and this also results in that the protein is no longer expressed. If you then design an antisense oligonucleotide to bind or to hide this intronic variant, you can restore normal protein expression. And this is the first application that we want to use for our DCRT, because this really, on a biological level, you will restore the normal protein expression, while in the other approaches, you will get a truncated protein that you then have to prove that it's still functional, or it still maintains as much as possible its wild type function. While with this approach, we are just restoring the normal protein function. So the chances that this will have a beneficial effect are much higher. Okay, as I said before, I'm just going to highlight a little bit the work that we've done on spinocerebellar ataxia type 3. So it's an autosomal dominant disorder. It's with a prevalence of 1 in 100,000. Onset is usually in midlife. It's a neurodegenerative disorder, and here you can see a healthy individual. And in SCA3, you can really see the ventricle is enlarged, and there is neurodegeneration in the cerebellum, but certainly also the brainstem is involved in SCA3. Symptoms are ataxia, paralysis, and distal muscular atrophy. And the cause is a CAG repeat expansion in the ataxin-3 gene. And CAG repeat, the CAG encodes for a glutamine amino acid. So it's a polyglutamine disease. And the polyglutamine expansion is in the ataxin-3 gene. The polyglutamine expansion is in the ataxin-3 protein. The CAG repeat expansion is in the ataxin-3 gene. And so this is the toxic gain of a function variant that I mentioned earlier. So really the idea about the splice modulation is that a mutant gene is expressed. There is in SCA3, there is an RNA toxicity reported as well, but it's translated into this polyQ expanded protein that is then cleaved. And we know that these N-terminal polyQ protein fragments are more toxic than the full-length protein. And these fragments really disturb quite a lot of cellular processes. And a hallmark of these disorders is that that in the brains of these patients, you will get these protein aggregates, both in the nucleus and in the cytoplasm. And by modifying the protein at this level, we hope to then reduce all the downstream toxic effects of this disease. So when you do splice modulation in such a manner, you always have to make sure that you know where the important protein domains are and what the main function of this protein is. So ataxin-3 is mainly involved in proteasomal protein degradation. It binds ubiquitin chains that are then cleaved by the Josephine domain. It has many, many different protein interactions and is involved in transcriptional activity. And we assume it's essential for cellular function. So that is an argument why we are not pursuing a gap-mer strategy, because that you would reduce the protein expression. We feel that these proteins are very important for normal brain function. Thereby, we would like to maintain 100% of their protein expression, but we want to modify them to make them less toxic. And so the CAG repeat is encoded in exon 10 of ataxin-3. If we design an AON that hides exon 10 from the splicing machinery, exon 10 is skipped. And this is a bit of an atypical example because actually the skip is out of frame. But because right in exon 11, there is an in-frame stop codon, we actually make a truncated ataxin-3 protein. And these ubiquitin interacting motifs are important for binding these ubiquitin chains. We still actually have 2 of these ubiquitin interacting motifs, and the entire Josephine domain is also still in this truncated protein, but it lacks the polyQ stretch. And then, so first proof of concept, we've designed many rounds of AONs. This is just a selection of 4 of them. We first always test them in fibroblast cells where we add the different AONs, and then we look for an extra shorter band on RNA, which means this is the exon skipped fragment. And then we also look on protein level whether this actually results in formation of a truncated protein. Out of these fibroblast experiments, we choose our AON that is most efficient. And but first we did some functional studies. So as I said, there are 3 ubiquitin interacting motifs that can bind to these ubiquitin chains that are then cleaved by the Josephine domain. And we wanted to investigate if this was a function, so the binding of these ubiquitin chains, if this function was still present in our truncated ataxin-3 protein. So here we made 4 different ataxin-3 constructs that were all labeled with GFP. So they all contain the Josephine domain, but this is the wild type ataxin-3 with 10 glutamines. Here is the mutant with 71 glutamines. Here is our truncated HXN3 protein that we produce with our exon skipping approach. And we've made a control construct where all 3 of the ubiquitin interacting motifs were deleted. We tested them in a U2OS cell line where there was a leg operator where this RNF8 construct could bind. And this locally on one single spot in the nucleus of these cells would introduce ubiquitin chains. And so wherever you see an M-cherry red spot in the nucleus of these cells, we know that there are ubiquitin chains incorporated. And then we transfect our GFP-labeled HXN3 constructs, and we actually measure the fluorescence in this one spot in the nucleus. And what you can see is that both the wild type and the mutant HXN3 efficiently bind these ubiquitin chains, comparable to what our exon skipped HXN3 construct does. And when we delete all of our ubiquitin interacting motifs, we see a significant reduction in the binding. There still is some binding because we know HXN3 homodimerizes, so there is still some wild type HXN3 floating around that binds to this spot, but mostly you can see a significant reduction. So that is kind of proof that we do not disrupt at least this important ubiquitin binding function of HXN3. Next, we went to a study in mice. And so for that, we used the MJD84.2 mouse model, where we injected our AON in the ventricles of these mice. So we would put a cannula on top, and we would do 3 500 microgram repeat injections into the ventricle. And then this is a mice that was sacrificed 2.5 months after our latest AON injection. And we stained for the backbone of our antibody. So this is a MOE fully phosphorothioated modified AON. And you can see that even after 2.5 months throughout the brain, you can really see that even in the cerebellum and the brainstem, we still have high concentrations of these AON, and that is compared to the PBS-treated animals. So then these mice, unfortunately, they do not have an ataxic phenotype. They're heterozygous mice. We are currently performing a study, also a treatment study, but then in homozygous mice, where hopefully we will be able to measure an effect on behavior as well. But we've only looked at molecular readouts. And here you can see that this is a wild type mouse. This is a transgenic mouse that's not treated. And when it's treated with our AON, you can see that there is on RNA level, there is a high level of exon skipping. And this is on protein. You can also see that there is a modified protein in this treated mouse. So in the brainstem, which is relatively further removed from the injection site, and in the cortex. And here is the quantification. So we get up to 40% protein modification 2.5 months after our latest AON injection. These mice also do not have protein aggregates as such in the brain, but you can measure insoluble protein with a dot plot. So that is what we did here. So wild type mice have no insoluble protein. These are PBS treated mice. This is a scrambled AON that we took along, and you can see that with our HXN3 AON that we really get a reduction in insoluble protein levels in the brains of these mice, also again in all the different brain regions. Another main feature is because we didn't get behavior and we didn't get protein aggregation. Normally HXN3 is localized in the cytoplasm, and we know if it translocates into the nucleus, it's a sign of toxicity. So here we quantified the nuclear localization of our HXN3 protein. So we stained for HXN3. And again, this is the mice treated with the control AON. If we then treat it with our HXN3 AON, you can appreciate that there is a reduction of nuclear localization of our HXN3 protein. So the different diseases have given us experience with designing these antisense oligonucleotides, testing them, and then also developing assays to monitor their efficacy. So we want to apply this in the DCRT. And our colleagues in Radboud, they focus on eye disorders and splice modulating therapies for eye disorders. We focus on brain disorders. And why those two organs? Because we can treat locally. That means you only need to administer a low dose and you get a high local exposure. The systemic exposure is low, so there is less risk for the kidney and liver, and you can have a lower treatment frequency than if you would with systemic delivery. So for which patients is this bespoke RNA therapy suitable? The mutations must be very rare and close to unique. We are currently not quite sure what that means. So we are using an arbitrary number of 5 patients worldwide, but we don't really know if that is the correct number, or maybe if there are 10 patients worldwide. We can also develop therapies for these people in the Netherlands, but for now we use the number 5. We are looking for mutations there where exon skipping can restore protein function in the brain or an eye disorder. It's important that the patient must benefit from the treatment. If if it's an eye disorder and all vision is lost, then that would not be a suitable patient for to develop a treatment for. And of course, the patient must be willing to undergo this experimental treatment, because it will always be an experimental treatment. What we've come to realize in the past year and a half is that European laws and regulations differ from that in the United States. So in the Netherlands, we can use the named patient setting, and it's called an artsenverklaring. And that does not require any regulatory approval. It's the neurologist or the clinician that will say, I think that this patient would benefit from an antisense oligonucleotide therapy, and it's the full responsibility of the neurologist. So all there are no safety rules or regulations that we have to abide by. We have to get local approval from the ethical committee, of course, but it's not like with the FDA where you can file for an IND. It's not like that in Europe. We do communicate with the EMA and also the Dutch regulatory agencies to advise us what is enough, how many safety studies and toxicity studies do we need to do to get the maximum evidence that we feel that the antisense oligonucleotides that we have designed is safe. The hospital pharmacy, they can prepare this GMP compound, but we are also are discussing whether maybe we can use a GMP-like compound. We would like to do the preclinical studies and safety studies in vitro as much as possible in-house. Here at the Leiden University Medical Center, we have an academic pharma platform where our animal facilities are trying to set up GLP-like conditions so we can actually do some of the in vivo studies in-house. And that really makes a difference for the cost of the development of these antisense oligonucleotide treatments. For now, the university medical centers involved are funding the therapy development, but the idea is once we have treated a few patients, we kind of know, or we can predict the developmental cost and the treatment cost over a period of years for each of the different patients, because the patients and the mutations are unique, but we think that the developmental cost for each antisense oligonucleotide will be quite similar. And if we know that, we can also talk about reimbursement through the health system in the Netherlands. So we are also having discussions with the Health Institute of the Netherlands. So for now, our preliminary timeline and sort of cost estimate that we have is that within the first 6 months, we will be able to design and test the antisense oligonucleotide in the lab, in cells, patient-derived cells or other cells. We can then do the functional studies in the next 6 months to test whether we actually do restore protein expression, and if there is a restoration of the function of this protein. Then we would like to do the safety and pharmacology studies in-house here at the animal centers of the medical centers. That will also take us 6 months, and then we can go ahead and start treating the patients. And I think this is a very, there are many initiatives to sort of align all the local and national initiatives worldwide. And I think that is essential if we're talking about these unique patients, that we do not try and invent the wheel separately, but we try and coordinate it as much as possible. And if there is another patient somewhere in the world with this particular very rare mutation, that we can use all the knowledge that we have in development of these treatments. Like I said, we're aligning with different initiatives, of course, initiatives at the OTS, but also within Europe and worldwide. So if you're interested, you can use this QR code to scan to get to this document, the briefing document. And I think in the last year, it's always a surprise in Europe that there are European laws, and then every single country in the European Union has their own little, puts their own little sauce on top of the European laws. And so we've been talking to our colleagues in Germany that are also involved in the worldwide initiatives, and already between Germany and the Netherlands, there are quite a few differences in the law that would really make a difference if we want to apply these N-of-1 therapies in the different European countries. So together we are trying to reach out to other countries within Europe so we can actually make a coordinated effort within Europe and standardize as much as possible. So where are we now on the basis of the regulation? So we're meeting with the European Medicine Agency as part of their innovation task force to get advice on appropriate in vitro toxicity studies and in vivo safety studies. We have another follow-up scientific advice meeting with the EMA in 2022. And once we know once we have developed our first therapies, we will discuss reimbursement strategies with the Health Institute of the Netherlands. Where are we based on research? Because I think the first year of the DCRT, we've only been spending our time talking, well, it was a COVID lockdown, so we couldn't do a lot of lab work, but we've been spending a lot of time trying to figure out how we should do it, organize it ethically, legally. But we are now also advancing with our first patients. So we are studying 2 patients with BPAN, that is an iron accumulation disease with mutations in the WDI45 gene. We have designed our AONs, and we are currently testing them in vitro, and I'll show a little bit about that later. And Radboud UMC is studying 1 patient with Stargardt disease, and there are 12 more patients that have been identified. So first I'll talk a little bit about BPAN. We are doing this together with our colleagues at the University of Tübingen. Oh, the light goes out. So my assistant will now turn the light on again. So BPAN, it's a beta propeller protein-associated neurodegeneration. It's a mutation in the WP4 protein that is essential in autophagy. It's characterized by an early developmental delay, and the main characteristic is on MRI level is iron accumulation in the basal ganglia. So actually here, this is the mutation in this particular patient. It's a deep intronic mutation, and this mutation causes the 100 base pair of the intron is included in the mRNA, and exon 4 is excluded from the mRNA. And this mRNA is out of frame, and so there is no protein expressed. So here you can see the effect of the mutation, the exclusion of exon 4 and the inclusion of this 100 base pair intronic sequence. And what we aim to do is develop an AON that covers this mutation, so we restore the normal splicing and normal protein expression. And like I said before, WP4, or the protein product from this gene, WDR45, is important in this process of autophagy, and autophagosome formation requires both WP3 and WP4. So we can use this autophagy as an assay to see if protein restoration actually also restores this important function. So we have designed 2 rounds of antisense oligonucleotide. So here is exon 4. This is the cryptic exon, and we designed as antisense oligonucleotides along these sequences. And this is one of the plots that we've done. But the primer pair that we've used specifically amplifies this cryptic product. And you can see that here, that is this band. So this is a cell line from someone, an unaffected a person. Here is the cell line from an untreated, from our patient with a deep intronic mutation. We added cyclohexamide into, so this is the lanes, the minus lanes are the lanes who are not treated with cyclohexamide. And these are the lanes treated with cyclohexamide. You can see the RNA is unstable when this cryptic exon is included. And here are 2 AONs that we've tried, the 4.8 and the 4.2, that didn't really have an effect on this cryptic product. But here, AON 4.7, you can see that nearly all of the cryptic product has disappeared. There is no antibody available for this protein, so we are now looking at autophagy assays to prove that there is a restoration of function. So the next step, we are looking for autophagy assays to optimize the concentration. Then we want to do some safety studies in iPS cells, and we are still debating on which in vivo studies we need to do. Then, of course, we need to look at the patient again, make sure that the patient is eligible, and if they approve, if they agree to participate in this experimental treatment, and the natural history study of this patient will then become important. And yeah, this is what we are still discussing. This is also something that will come back in the eye disorder that I'm going to discuss. We are now really at the stage where we need to really decide what we want to do for our tox and safety studies. So this is Rob Collin here from Radboud. He kindly gave me some of his slides to discuss his AON development for Stargardt disease. So these inherited retinal diseases, they actually show quite a lot of clinical heterogeneity, but also a genetic heterogeneity. And there are a lot of different mutations in hundreds of different genes that can underlie inherited retinal diseases. So Stargardt disease, the clinical features of Stargardt disease is that it's a progressive disorder, mainly affecting central vision. It shows a variable age of onset and disease course, and it's characterized by flecks on autofluorescent imaging. And it's caused by biallelic mutations in the ABCA4 gene. And really, even if there is a clinical, is there variability in the clinical characteristics, really the aim of an AON therapy would be to delay a disease progression and maybe even disease onset. Stargardt disease 1 is a progressive IID, and it's exclusively caused by ABCA4 mutations. ABCA4 protein dysfunction leads to accumulation of lipofuscin, which is a waste product. So the patient that the DCRT is focusing on for Stargardt disease is a male of 37 years of age with a biallelic mutation in ABCA4. And it's actually this deep intronic mutation that is targeted by the AON to restore protein expression. So the group of Rob has made a mini gene with the variant of interest inserted. They transfect the mini gene into hex cells and then look with RT-PCR analysis to see if they can restore the correct transcript. So here are the ASOs that were designed in Rob's group. And then these are wild type cells. These are the wild type MIDI gene. This is the mutant MIDI gene where the different AONs were added. And you can see that in 2 of the AONs, the correct transcript was formed. And the same AONs were transfected in fibroblast cell lines from a different patient than the patient that I showed you before. But also there you can see addition of cyclohexamide stabilizes the transcript, and then you can see the pseudoexon and the correct transcript, but addition of these AONs then make all the splicing correct, the normal splicing. So again, they had a meeting with the patient to inform the patient about the study and to collect cells from this patient to do these in vitro assays on this patient's cells. So the molecular work entails generating the cell lines, order these 5 AONs and test them in this MIDI gene system and fibroblasts, optimize concentration and safety in pharmacology studies. The patient is being seen now for a natural history study, which entails an in-depth clinical assessment every 3 months, and the discussions with the regulatory agencies that are up and coming. So in the Netherlands and maybe in the LUMC, we really use a lot of exome sequencing for diagnostics. And I think quite a lot of the mutations that we are interested in the DCRT, we are actually not looking for because we're not looking in the right spot. And I think there are quite a few patients that are not diagnosed. They know it's a genetic, their disease has a genetic cause, but the cause has not been identified because we're mainly still looking at exons and not just at introns. So I actually think it's underreported, the number of patients that we might be able to help with these N-of-1 therapies. And so if anyone has identified interesting mutations with an eligible patient, we are interested. If you are interested in the DCRT, we have a website, so you can have a look, or you can email us, of course. And these are the people that have been involved in the research. And with that, I would like to thank you for your attention.
What a wonderful talk. I have to thank you for providing the perspective of kind of the whole story of your research and not just the scientific ideas. It's really helpful, especially to trainees, to really kind of realize and think about from these perspectives how we're moving forward. So I really enjoyed that portion. I'm going to turn things over to Ronald now, who will moderate the question period. I see there's been several great questions in the chat, so we're looking forward to that.
Yes, it was a great presentation. So I will start with a question from my side. So I was wondering, how do you select which patients are eligible for these kind of treatments? Because yeah, you already mentioned it. These are very rare patients with very rare diseases. Only 1 or 2 patients are known. So probably you need to have a little bit of insight of what kind of mutation it is, what kind of exon is skipped, maybe a little bit of functional assay. So probably this will take a lot of work. So how do you select the patients eligible for?
Well, selecting or finding them, it's like, and also when there is a mutation that is eligible, is then the patient also eligible? So if the disease, if it's a slow progressing disease, probably not, because then if we would have to treat a patient for 5 years to determine whether our AON has a clinical benefit, that is probably too long. Maybe all the important functions are already lost, and I think it's safe to say that we cannot restore function that is lost. If there are brain cells that have died, we will not get them back. So there are not a lot of, it's not so much as selecting a patient. We can select a mutation for sure, and we are quite strict now because we are aware that we have to select the most optimal mutation with the most optimal patient to show that we can do it. Then I think it's just a matter of looking at the RNA. If it's one of those deep intronic mutations, does it induce actually an inclusion of a cryptic exon? If it's an out-of-frame mutation in a coding exon, is it a big exon, small exon? What protein domain does it encode for? If it's, for instance, a part of a DNA binding domain, and you have one amino acid that is missing and it will disrupt this DNA binding function, skipping an exon will not improve it. We've had very interesting mutations in genes that just had one exon, so that is an easy evaluation. But it's manual, I think. I think we assess the mutations one by one, and then we need to assess whether it's suitable or not. So it's mutation, and then it's patient, and then we need to see if the AONs that we have designed are efficient enough and safe enough.
Yeah, and maybe to follow up on this question. So I think when you look at the clinical trials for RNA therapies, it's for medical centers, it's really difficult to include more than 3, 4, or 5 patients in a clinical study in one center. So how do you envision this? So how easy is it to because it's a great strategy, and probably many patients can benefit from this. But do you think that the medical centers can cope with the number of patients and the number of therapies that you can develop with this strategy?
I think, for one, there will never be hundreds of patients. I think that we will be treating at the LUMC, for instance. There will only be a few, and I think the clinical trials have a totally different design, also in how they study what type of tests they do with these patients, clinical assessments, and I don't know. We will have to wait and see if it will be as intense as it is for the clinical trials that are currently up and running. But I mean, we're talking about ultra rare diseases, so ultra rare patients, and I think what we want to do as university medical centers is help people. And I'm not saying that we want to treat all the Dutch patients at the LUMC. We want to help people develop these therapies, and then they can be treated at their local hospital. It doesn't have to. And then I think I definitely think it will be doable. But we want to put up this kind of support network with developing these AONs, but also the clinicians. You might, for each patient is unique, but the function that that you want to restore, or maybe all the ataxia patients, they probably can be assessed by quite similar clinical assessments. So we also want to set up this whole network of neurologists to kind of coordinate what we will measure, how we will measure it, and when we decide it's not beneficial or it's not safe, so that is what we want to provide in this network. But we certainly don't want to treat all of the patients in only a few centers. I think that can be spread throughout Europe. That is the idea. And yeah, it's ultra rare, so we will not. It's not like we will have 100 patients that will come in. But you're quite right. It's quite labor intensive to treat these patients.
Yeah, they are ultra rare, but if you sum up all the ultra rare patients, it's actually just a common disease, right? The incidence of ultra rare diseases.
But I think also we do not have to. We are not doing a clinical trial, so we do not have to have that much proof on safety. And I think it's an assessment whether it's not detrimental to the patient, and if we can see that there is a difference in disease progression. And I don't think we have to, like a proper phase 1, 2A or 2B clinical trial has got a whole battery of safety parameters that you need to do. I think that is not required. I'm not saying that we don't want to do any of them, but it's not as extensive as it would be for a clinical trial, because we will not do a clinical trial and we will not go for drug registration.
Yeah, clear. Thank you. Then we go to the questions in the chat. So if you have any questions, please use the chat function. And we already have quite some questions. So the first one is from Claire. What methods do you use to measure the exposure of the AONs?
To measure the exposure of the AONs?
Sure what you mean.
No, I'm not quite sure what you mean.
But I guess, how do you measure the effect of the AON? So do you have like a molecular readout or a functional readout?
So that is very, and so this is also why we want to have a large network, because every protein and every pathway that this protein is involved with will have their own particular assays. And so if we get a wide network of people that use that have an expertise in different diseases, they will already have ready-made assays for autophagy, for protein aggregation, for mitochondrial function, for I don't know what there are different assays. So we don't have to start from scratch designing these. And we mainly efficacy, we just use it in the level of RNA and protein modification. And maybe you were asking a question about how we measure if we have enough RNA in our cells?
No, I think maybe the question can also be like, how do we measure the concentration of the AON in the target tissue?
Yeah.
So are there methods to do this?
Yeah. So there are methods to do this. So you can do this with an ELISA, but there's now also a publication that you can do it with qPCR. So we are also looking into that. In our animal studies, we just do an immuno with this backbone antibody, and we have done some SPECT radio-labeled AON studies where we look at how it distributes throughout the mouse brain after injection. So you can do it in different ways, but yeah, what we want to do is we want to modify protein expression. So we just look at how much modified protein we have in the tissues that we are targeting.
Yeah.
I think that is the easiest way to do it. Yeah.
Yeah. Hello, Wilke. Nice talk. Can you share your opinion on the selection of cell lines based on the target analyzed for ASO inhibition? Do you recommend some cell lines to be better than others for ASO transfection and downstream analysis? So what kind of cell lines do you want to use?
I think the main thing is the RNA that you are targeting, is that expressed in that cell line? And then is it a mutation specific? Because these cryptic splice mutations, you really need to have a patient cell line, because there is only aberrant splicing in this patient cell line. So for that, you would need some type of patient-derived cell. To be really honest, for optimization, so the first cycle of designing these AONs and testing them, we really standardly use fibroblast, controlled fibroblast or patient fibroblast. But we've also made the mistake that we thought our PCR wasn't working, and then when we looked, it turned out that this RNA was not expressed in fibroblasts. So you do need to check that. And then if that's not the case, you need to look for another cell line. And we now we can go into iPS cells, look if it's expressed in iPS cells, and if that's not the case, you really need to do sort of a tissue-specific differentiation. So, but I think an easy cell line, especially in the first stages where you are interested in, is it working and which AON is working better than the other one, then you can really, it doesn't really matter which cell line you do, because we do know if we pick the least efficient AON if they're in fibroblast cells. In our experiments, we test 4 AONs, and AON 4 is the most efficient and AON 2 is the least efficient. If we take that into other models, that will always be the same. Then always it's not like suddenly AON3 is more efficient if you inject it in the mouse brain. That's really, so what you find is quite in the fibroblast or a neuroblastoma cell. You can also use neuroblastoma cells. It doesn't really matter. If you want to decide which one is more efficient, then but then how efficient is it? How much protein modification do I get? There you either need to wait long, because then it makes difference if you transfecting, you wait for a week, or you wait 2 weeks, or you inject it in a mouse, and then you always have 60%. You usually have way more protein modification in your cells than if you inject it in a mouse. So you cannot say something about absolute numbers of protein modification based on your cell model, but you can choose your AONs, which one is which one is inducing the splice modulation that I want. And if you have 5 AONs, you can select the one that is most efficient.
Yeah, so I think the answer is to start with the easiest cell line you can find, where the
check whether your RNA is expressed. Top tip.
Thank you. Then I have a question from Georgia, and the question is: are you? I think you mentioned during your talk, you mentioned the work on SCA3, but are you also interested in strong cognitive disorders?
Strong cognitive disorders. Yeah. So I'm in my group. We're working on a point mutation in the APP protein that causes cerebral amyloid angiopathy. So amyloid accumulations around the blood vessels that have dementia also is a phenotype that would, in principle, also be applicable to Alzheimer's disease, because it prevents the amyloid from being formed. So you get a reduction in amyloid peptide formation. But I already have too many diseases that I'm studying in my group. So I'm very interested, but I'm not studying them at the moment. No.
Are they of interest for the DCRT if you have a very rare patient?
If that, well, the thing is that I think the thing you need to ask yourself is it a rapidly progressing disease, and can I measure it? Is there any outcome measure that will show me that the disease is progressing? And if there is a very fast cognitive decline where you have a reliable outcome measure for in the clinic, because that is what you need to do. Eventually, you can do a lot in cells, but you need to, from the moment you start treating a patient, you need to measure something to say that we are actually making a difference and a positive difference in this patient's clinical phenotype.
Then we have a question from Lisa. With no history of safety efficacy in NS1, can you talk about the informed consent process and the involvement of ethics committees?
So ethical approval will always be essential, so also locally. But we also really, so in this whole European setting, a very important part is also the ethical part. There is a legal part, there is an ethical part. When do you ask a patient if they want to be part of this experimental treatment? Are you first developing antisense oligonucleotides and then you ask, but then the patient might say no, and then you've already put in a lot of effort into making an antisense oligonucleotides. Do you ask them first, but maybe then you cannot find an antisense oligonucleotide that actually can help this patient, or maybe it can correct the splicing, but you cannot make an antisense oligonucleotide that is safe enough. So I think these are also, and you know, if it's a young patient, you know, how sick, it's also always a little bit crude, but how sick can a patient be before you can do an experimental treatment? If the disease doesn't have a very high impact, then the risk of developing such an experimental treatment might actually be higher than, you know, the risk of the disease progressing. So it's a very good question, and I think this is really where we need to be very careful and yeah, that's what we're trying to do. But I'm probably in 5 years' time, I'll be able to answer that question a little bit better.
Okay, then I think the next one is from Elisa. I think it's a straightforward question, might be, but maybe not. Since I think all the very nice work from Tim Yu on the Milasen and the acceptance and all the work on the RNA therapeutics for COVID, do you see personalized ASOs becoming more common in the coming years?
I think they should, but we still have to, I think we just need more patients treated, and the development has to be faster, and I think we need to figure out what is enough safety data. So I think it's too early to say that. I would hope that would be the case, because really this is a group of patients that so far have not had any outlook on any type of treatment. But yeah, I think we can do it, but we still have to do it consistently. Yeah.
Then we have a question from Alex, and the question is, do you think that molecular work is enough just to see an effect on RNA or protein level, or do you think that you also should do any functional assays, both for brain and for eye diseases?
Yeah, I think it's a very important question. I think that is the reason why in the DCRT we have chosen now for restoring normal protein expression. So if there is no protein at all, but you restore the normal protein expression, the chances that that will be beneficial and not detrimental is quite high. I think for the scar example that I showed you, where you remove a part of the protein to make it less toxic, you end up with a truncated protein. And I think then biologically you need to answer a few questions, whether you need to know what the most important function is of this protein. Then you need to have an assay where you can test this function, and you need to see that after your treatment, the function is restored. So I think it really depends on the mutation and the type of splice modulation that you are trying to develop.
We have a question, actually a remark from Louisa. So she's considering applying for a PhD vacancy in your group. So that's good news. And the question is, what are the risks associated with these therapies identified or predicted so far? So are there any risks on ASO therapeutics?
Absolutely, yeah. So one, you have the intrathecal delivery. So that is not, we envision now like every 3 or 4 months. There is a GAPMER antisense oligonucleotide trial in Huntington's disease that was halted because of side effects. It was quite a high dose that these patients received, but the highest dose group actually showed a worse disease progression than the placebo group. So there were enlarged ventricles and worse clinical scores. We don't really know what happened yet. So all of the biosamples are being collected. The dosing has halted, but the patients are still coming in to the clinics to see also for the washout period. So we don't know what happened there. But again, that was a GAPMER AON, and we are making use of a different chemistry that has been used in nusinersen, which has been, patients have been treated with that type of modification for a long time. So we don't really know is the real answer. But again, that is why in the DCRT, we have experience with designing these splice modulating ASOs, because that is actually not as straightforward as it looks, because you only have a limited amount of sort of space on your transcript where you can target the AON if you want to induce this splice modulating effect. So I think for these fully PS backbone MOE modified ASOs, the safety profile we think is more, it's probably more, we have more knowledge on that. But yeah, also here the future will tell, and we just need to monitor carefully.
Thank you. Yeah, so it's 6 p.m. here, so it should last 1 hour. To me, it's fine if we spend because there are a lot of extra questions. So is it okay if we do like 10 minutes extra to answer some more questions?
Yeah, and I can also go through the chat and answer people there. I don't know if they still have access to it, but
yeah,
Great,
we can.
Let's keep asking questions. There will be a recording, so if you have to leave on the hour, that's fine. But if Dr. Van Roonmam is able to stay, let's keep
that's fine. Yeah.
Cool. Then we discontinue, and because there are still 11 or 12 questions on the list. So thank you again from Monica. For how many mutations or patients can you perform research or develop a treatment per year?
So are we talking COVID measures? Because then we can do a lot. If you give us a lot of money, we can do it for quite a lot of diseases. Yeah, I don't know. Ronald, how long does it take? It's just, I think, within 6 months with a full-time technician, I think you can suss out which ASO is most efficient in doing your splice modulation. That is, so that goes from sitting behind your computer, designing the antisense oligonucleotides, order 10 of them, figuring out which ones is the most efficient. You can even do a second round of, so if you had 10 people, you could do 10 if you have 100 people, you can do 100 we don't have that number of people, so but it's a matter of just hands and brains thinking and doing.
Then a question from Peter. Interesting talk. Thanks for taking the time to present. You mentioned that you are engaging with the AMA. Can you share what your impression is about their interest in supporting getting a good process in place for handling these type of activities? So how's the contact with the EMA?
They're very supportive. I think everyone is very supportive. So I think what we've learned in the last year and a half is that every institution or organization that we've asked, where we went to ask our questions, no one really had an answer for us because there is nothing in place for a thing like this. But at the same time, they're all very willing and very supportive to help us develop this. So we still don't know exactly how it's gonna go and how we're gonna, but everyone is very willing to think with us. And so also with the EMA, they said, if you just outline all of your questions, we can invite experts that have been involved in other clinical trials with other antisense therapies for different diseases, not just brain or eye diseases, but they can help us set up in vitro assays that maybe are a good addition because we don't have to go into non-human primates or other bigger models, and so they will help us set up a comprehensive set of toxin safety studies, hopefully to predict if our antisense oligonucleotides is safe enough. But also the Dutch health ministry is very supportive in helping us, and the Health Institute, the Netherlands, and even the health insurance companies are interested. So I think it's really indicating that this is a need that needs to be filled, and people are supportive.
Yeah, and how is it? You need to go to every country to have you have to need specific different regulations.
Also there, as we go along, we figure out because our colleagues so here it's the named patient paradigm, and apparently in Germany it falls under the criminal law. So there is a slight but important difference there. And how it is in France, it's different in France again, but how specifically different these are all things that we are figuring out at the moment.
Yeah, it might be nice if they all align and if you can have one.
Yeah, or at least if we know what the differences are per country, that if we have, it might be that there is an AON designed in the US and there turns out to be a patient in France, then at least if we already know the differences between the different countries in Europe, we can advise as well. It's gathering information to questions that no one has asked before, but at least if we're asking and we're getting an answer, we want to provide it as a resource for the next question that will for sure come.
Next question from Mayon. So is there anything known about the disease course of these individual patients? So are you doing any natural history studies?
So the idea is that what we're working on now, and what is also being done in the States, is that from the moment that a patient is identified, that will be the start of their own natural history. So then they come. So for Radboud, they're seeing these patients now every so many months to kind of get a progression in the normal history of this one particular patient. And then from the moment you will treat this patient, you can see whether the decline in vision will be the same, or it will be less, or if it's more, then we're doing harm. So that is, we're trying to get them a natural history profile in the period that we need to design and test this antisense oligonucleotide. That will take about a year and a half. And then we can use that year and a half to sort of get a natural history study of this particular patient.
Okay, and then the question about this is, how will you know what the efficacy will be on the AONs in the patients once you will be able to treat them?
We don't know. No, so we can only measure. I mean, it would be nice if we would have some type of, if we can measure in CSF, if we can measure the protein in CSF, or, but I think for now we are aiming for clinical improvement. Or not clinical improvement, at least a halt in disease progression, and no overt signs of a detrimental side effects. Yeah.
Clear. For the ultra rare patients with the non-degenerative diseases, how long do you take treatment will be? Because probably, I guess it will be a lifetime treatment.
Yeah. It will be a lifelong treatment.
So is it supported by the DCRT? Do people get a lifetime treatment, or do they get
treatment
for 2 years? Or how do you...
Well, manufacturing an antisense oligonucleotide with these local deliveries, I think that is important to say. That is also why we've chosen these local deliveries, because you need less antisense oligonucleotides, and then it's not that expensive. You can calculate how much antisense oligonucleotides you would need if you would have like 4 injections per year or 3 injections per year. You can sort of calculate what you would need over a lifetime, and that is actually not a lot of costs. So, but then there are interesting discussions because you have GMP, a GMP batch or a GMP-like batch. The GMP batch is more expensive. It also takes longer to produce. So, then we would also need to wait longer before we could start treatment. And so that is also discussions that we are having. Are we allowed to use GMP-like antisense oligonucleotides to treat these patients with, or do we need GMP?
Yeah, and is there anything known about the time interval? So should you treat the patients in the beginning of the treatment every 2 or 3 months, and can you have a longer time interval between the different treatments?
Yeah, so I think for that we really do copy paste from the trials that are ongoing at the moment with antisense oligonucleotides, and the dosing that has been done also in the few patients in the States that have been treated with these antisense oligonucleotides. So.
Okay, so Annemieke mentioned, and I think you mentioned it before, that there are 2 PhD vacancies in Annemieke and Willike's group.
Yes,
so,
if you are willing to work for Annemieke and me, then you are welcome. I think I already saw Annemieke paste the link in the... Yeah. And you can ask, when we're not around, you can ask Ronald if it's a good idea, if you want to apply.
Yeah, and be aware that you have to sit with a blue thing next to you.
That's your own choice.
Well, it's not. So, following question. Could you elaborate a bit more on what you thought when you read the article from Tim Yu on Milasen?
Oh, the first and only thing that we said really is we can do this. So because that's something Annemieke and I have been doing for the last decades, designing these AONs, testing them in cells, testing them in animal models, and then we would hand over to biotech companies, because for the clinical studies, that's really not feasible in an academic setting. So if you take it out of the clinical setting up for clinical studies, and then it's doable also within an academic setting. And if you can do it within an academic setting, you can also keep the cost low, and then it's also more affordable for. And so I think in the Netherlands, that would also mean that reimbursement would be much more feasible for a patient who would otherwise would not have any treatment, and then would rely more on the health system because of their progressive nature of their disease. So I think there's also a cost benefit there. Because we do not want to file patents, or that is also quite people forget, but actually having a patent and filing it and maintaining it for an academic institution, that is actually quite expensive. We don't need this. It's one patient, and if it turns out that we've designed an AON and a company is interested in it, we can always file for a patent afterwards. But the whole patenting process is not part of an N of one therapy development. So I think doing it like that, if we can do it in a safe manner, I think that is something that we are really focusing on now. How can we do it within an academic setting, within a safe manner, so we can do it fast and we can do it safe, I think. And then it will be affordable, and then it can be reimbursable. And then I think we have a sustainable model.
Okay, we have 2 minutes to wrap up. So and 2 questions. So if you can give 2 very short answers. So what are the chances of inducing immune response?
Low, yeah, but I think for this type of chemical modification, it's low. Is that short enough?
It's short enough.
Okay.
And how is pharmaceutical or clinical quality oligo production is established for the lifespan of the treatment? So how do you get enough of the oligo to treat the patients for life?
Yeah. So now that a very good question, but can we, because the more you make, the cheaper it is, can we just get enough oligo to treat the patient for a lifetime? But if we dissolve a new batch every 5 years, is that batch then still as effective as the first batch? Or do we just need to order fresh oligo every year, but that would increase the price? Or it's a very good question. So if anyone knows the answer, they can email me. But we are also talking to European manufacturers, American manufacturers, if they can advise us. The pharmacy is important for this because they will be responsible for aliquoting it. Do we need to get the pharmacies to aliquot it and make it ready for injecting, or do we need to ask the manufacturing company to do that for us and then send us they're all it's very good questions that we are still working towards an answer.
Okay, now I can do this. One more question from a parent of a patient who asked whether he can contact you.
So that depends. You can always, first of all, you can always contact me. But second, we've had a lot of American and Canadian parents contact us, and it's just not feasible for a patient that is not, at least not a European patient, for us as the DCRT to be involved. So then we are happy to bring these patients and parents in contact with people that are more local, but you can always email. Feel free to email.
Okay, he's from Europe, so no, I think feel free to. I think Annemiek already. The email address is dcrt@lomc.nl, so you can always send an email.
Exactly.
Okay, so Erin, word to you.
Great. Well, this was an amazing seminar. Obviously, there was a ton of engagement, lots of questions. I think probably still some questions in some brains out there. And so you offered email as a way of communication. I see you're also on Twitter. So there's lots of ways to get in contact.
Yeah, but I wouldn't say that Twitter is a reliable communication with me. I don't use it a lot. Maybe yeah, but that's fine. Okay. Email is probably a safer way of communicating.
Perfect. Good to know. So make sure you're paying attention. Use email. But we want to thank you so much for this amazing seminar. It's really has been our pleasure. And I want to let everyone know who's still watching or who's watching the recording that we are planning more seminars. We should have a fun one in December, and then there'll be more in the new year. So please do keep an eye on the OTS social media pages, our website, and we are hoping to see you at the next session. Thank you so much, everyone.
And thank you for inviting me. Bye-bye.